Okay, now we are on tube now. So let me start with a brief introduction, and then we will start today's seminar. So today we are very happy to have Dr. Atril Mukhopadhyay from Ben's group. Dr. Atril Mukhopadhyay received his bachelor and master degree from the Indian Institute of Science Education and Research in 2014, and he received his PhD from the University of Hamburg in 2020. So after that, he worked as a postdoc in Dr. Ben's group at TU and Darmstadt in Germany. So his research interest focus on the active soft matter, the nonlinear dynamics, and the directed particle transport. So Atril, please go ahead, your talk.
Thanks for the introduction. So good morning, good afternoon, good evening to everybody joining today's talk. So I'll talk about our ongoing research where we develop a new type of droplet swimmer, which can show circular motion, a chiral circular motion, and this is induced by memory effects. So this work has been done in collaboration with our research group of Rand Neo in HUST China, who performed the experiments. And the numerical simulations were performed by us in Memory-induced group at TU Darmstadt. So for those of you who haven't been to Darmstadt yet, so we are located in the southern part of Germany, very close to Frankfurt. And Darmstadt is a home to two science universities, and there are many prominent research institutes and also industries. And our own institute, where I work, is in the Institute for Condensed Matter Physics, also known as IPKM in short in German. So we focus on both theoretical and experimental research on soft matter physics, but we have other systems and other research groups too as well, working on complex systems, biophysics, and hybrid quantum systems. So a brief overview of what I'm going to talk about today. So I'll start by providing a brief background to the field of active matter and why they are so interesting for researchers across disciplines. Next, I'll talk about the experiment performed by our collaborators where they observe chiral motion of the active droplets. And then I'll talk about the numerical simulations we performed in order to understand and explain this phenomena. And finally, I'll end with a summary and some concluding remarks. So let's get started. So what are active swimmers? So for physicists, active swimmers or active matter in general are particles which are capable of converting their internal energy, or they can consume energy from their immediate environment, and they can give rise to systematic movement. And they are found at all possible length scales in the universe, right from the cellular motors at cellular level to the bacteria in the microscopic length scales, and even up to our everyday lives, like in macroscopic organisms like fishes and birds in this macroscopic level. So why are they so fascinating, and why do we study them? So first of all, these systems can show very nice navigation properties to extremely complex environment. And these are important in order to perform many vital functions. For example, the molecular motors like kinesin in the human body can use the energy derived from ATP hydrolysis, etc., to move cellular organelles or cellular cargo in the human body on the cellular level. So this is required for many important body functions like protein synthesis, DNA replication, etc. Also, bacteria can sense the chemical gradients in their immediate environment and can navigate towards food or even away from poison or predators. Birds can also navigate like a pro in turbulent weather conditions using their wings. Another important aspect is that these agents can not only move on an individual level, but they can also show fantastic collective level dynamics, which is kind of an emergent behavior when they group together. So for example, as we have already maybe seen many times in many talks, so in the swarming of birds, so birds of same feather can actually flock together and exhibit very nice swarming behavior as shown in this figure here. And this can allow in the individual birds to escape from the predators by forming a spontaneous groups and then moving together in diverse patterns. A very recent example shown on the right side is for the starfish embryos. So during the development stage, these embryos can spontaneously form clusters, as you can see here. And these clusters can get arranged in a crystalline fashion. So, and they have unique properties like they can rotate and show collective chiral motion together. So in order to understand all these, how this living active matter can perform these complex tasks, like interacting with the environment and show this kind of fascinating collective dynamics, it is really important for us to study these systems. And this will probably allow us to also design synthetic active particle in the lab, which can be used for advanced technological applications like, for example, controlled drug delivery in the body, etc. So speaking of the artificial or synthetic active swimmers, there has already been considerable progress in designing these kind of swimmers in lab, and there's already been many types of active swimmers already synthesized. From the design perspectives, there are two key ingredients which set them apart from the passive swimmers, which shows Brownian motion. So firstly, they require a source of energy, which will fuel their movement or self-propulsion. And secondly, since the motion is itself active, and they should have a directionality of the movement. So the second requirement invariantly requires that some sort of symmetry breaking, which can either be spontaneous or can be explicit by design. So here I'll just try to show only two examples of many different types of from many different types of synthetic active particles. The first one is a Janus particle or a Janus swimmer, which has different chemical coatings on the two sides, on the two hemispheres. And this leads to different chemical reaction when it is immersed in a particular kind of solvent. And due to these chemical reaction occurring on the two different sides, they can, this acts as a fuel, and this particle can move along certain direction. So contrastingly, with these Janus particles, which are asymmetric by design, we also have droplet swimmers, which are basically liquid droplet immersed in another liquid. And these droplets are isotropic. So we don't have an inherent asymmetry or anisotropy in them. But they can spontaneously develop an asymmetry on the surface because of the surface tension gradient. And this leads to certain Marangoni flow or Marangoni stress on these droplets. And as a result, these droplets can self-propel. So in the course of the talk, I'll talk in much more details about these droplet swimmers. So not only linear self-propulsion, but also the natural active particles can propel or exhibit in a chiral fashion and show circular motion, which is again very important depending on at which living conditions is bacteria or the microparticles live in. For example, in some bacteria like Listeria, as shown here, they can form trails of actin molecules, and they can use these trails to move in a circular fashion, and this helps them to transmit or infect their cellular targets and also infect other new cells. Bacteria like E. coli can also take advantage of this kind of a circular motion to optimally cover surfaces, and this allows them a broad coverage area. So as compared to the linear movement only, they can cover a broad surface area, which is very important in the search of food or to even find a favorable living habitat. So based on these examples, so researchers have already designed a few chiral artificial swimmers. For example, we have Janus spheres, Janus silica colloids, which when half coated with carbon, they have been shown to show chiral motion in non-Newtonian viscoelastic fluid, as shown in the first diagram here. They tend to, it is also possible to have Janus rods, and they can also show circular movement when they're placed in a suitable fluid medium. And when this fluid medium are embedded with certain certain spherical obstacles. So they tend to move around these circles, around this obstacle, and due to the hydrodynamic interactions. when we place it in a particular fluid medium. However, when compared to the linear artificial swimmers, these techniques used to generate the chiral swimmers and the chiral motion in active swimmers are fairly limited. And moreover, the existing techniques have certain limitations. For example, they either require a special particle design like an asymmetric particle shape, or they need a specialized environment like viscoelastic media or a proximity to certain walls or solid interfaces. And this provides the motivation for our current project, which I'll talk about today. And here we try to address these limitations and develop a new class of droplet swimmers, which can propel without requiring a certain asymmetric particle shape or any specialized environment. So this brings us to our experimental setup. So this part has been done by our collaborators in China. So we have a droplet, a liquid droplet, which is composed of a highly alkaline mixture of two polyelectrolyte, that is PSS and PEI. And additionally, we have a solvent in the Petri dish, which is just an aqueous solution of hydrochloric acid. So what happens when we just drop one drop of these droplet on the solvent? So initially, as you can see, when the droplet is placed on the Petri dish, the droplet instantly moves towards the edge of the Petri dish, as you can see here in the video, and it starts revolving in circular orbits around the whole Petri dish. Okay, and the radius of this orbit is almost similar to that of the Petri dish. So this motion continues for about 200 seconds in the experiment, after which the droplet moves away from the Petri dish. And in these intermediate time scales, as you can see here, the droplet still shows circular motion. It is still moving in a circular orbit, but the radius of the orbit is much smaller. So it really moves away from the Petri dish boundary. And this change in the radius of the droplet is not due to any geometry of the setup, but it occurs spontaneously. You can also notice that the speed and the direction of rotation, which is now anti-clockwise, is also spontaneously chosen by the droplet, and it's not due to any external means. So these chiral oscillations in the intermediate time scales continue for long times, so about 200 to 300 seconds, and after which another transition occurs. So now you can see that the droplet is still showing circular motion, but it is confined to a very small area of the Petri dish near the center of the droplet. So the droplet is still showing a chiral motion, but is as if already trapped. And so the motion eventually comes to a standstill after long time scales, and the droplet eventually stops. So based on these the three dynamics of the droplet, we can actually characterize the whole dynamics in three different stages. The first one, where the droplet moves around the Petri dish, is a ballistic phase. And in the intermediate time scales, the motion is chiral, with smaller orbits, and this is we call the chiral phase. And at the very end, we have the self-trapping phase, where the droplet is confined to an extremely small region. How to characterize these three phases? We do it by considering the maximum displacement of the droplet in a certain time interval delta t. So the delta t is chosen to be the one oscillation period of the droplet in the self-trapping regime. And what we do is that we find the average displacement of the droplet in these time scales. So in the ballistic phase, so after normalization, you see that the droplet shows maximum displacement. So the delta r is close to 1. The value drops off to an intermediate value like 0.5 in the chiral phase. And in a self-trapping phase, the droplet hardly moves. So the maximum displacement is almost close to 0. Okay, so what is the mechanism or the physical mechanism, the process which leads to this kind of a droplet behavior? So first, the droplet starts to move, and it shows this kind of three dynamics. So what is the physical mechanism behind it? So what you see here is actual snapshot of the droplet taken at different times. So initially, you can see that the droplet looks extremely transparent because it's still liquid. And as time progresses, you see that there is a opaque region forming towards the edge of the droplet. And this is because the droplet, when it is placed in the solution or a solvent in the Petri dish, starts to solidify at the edge. And this proceeds, the solidification process proceeds very slowly towards the droplet center over time. So what is actually happening is the droplet is extremely alkaline, but when it's placed on the Petri dish liquid, the protons flow in the droplet and it leads to a complexation process, which enables the droplet to freeze or self-freeze at the outer boundary. We could actually confirm this by an FTIR absorption spectroscopy. So in the liquid phase, the droplet has a dominant NH2 NH2 contribution. The peaks come from this NH2 group. Whereas in the solid phase, it is the NH3 ions which leads to this absorbance dips. Now, what happens because of this slowly proceeding solidification at the droplet edges is that there is a small porous structure forming at the droplet boundary. So as you can see here, this droplet pores allow the liquid inside the droplet, which is still the liquid polymer-polymer mixture, to come out through this pores. And once this the polymers come out through the pore into the surface of the liquid in the Petri dish, it can reduce the surface tension of the fluid, and this eventually leads to the droplet motion. So we can characterize, we could measure the emission of the polymers from the droplet, and it shows, and they show an exponentially decaying behavior over time. And this polymer, they reduce the surface tension of the fluid, and this gives rise to Marangoni flow, as you can see in the middle panel. So these flows, we can measure the strength of these flows both away from the droplet and with time. And we see that the speed decreases as we go farther from the droplet, and also decreases with time. We can also measure the speed of the droplet over time. And we see that the droplet speed initially starts at around 4 centimeters per second, and slowly it drops down in magnitude over time. So this corresponds to roughly 10 times the body length of the droplet per second, which is extremely efficient for these kind of droplet swimmers. So how can we understand the process which leads to this movement? So how do we numerically understand these processes? So for these, to understand the mechanism of the motion of the droplet, we introduced this simulation model, which I'll explain in a bit details here. So we consider the droplet as a 2D disk. So although this is a 3D phenomena, we saw that qualitatively we can describe everything with the 2D model. So the droplet is considered like a spherical disk, which gives off the polyelectrolytes, which is accounted for by this exponentially decaying surface activity of the droplet. The polymers which come out from the droplet, they can diffuse in the fluid around in the Petri dish. And it is important to note that the polymers, they diffuse with a very low diffusion coefficient. And we will see that this is very important for the droplet motion and the memory effect, which comes later. So once the polymer is emitted, due to this polymer, the surface tension of the fluid is decreased, which we take into account through this kind of an empirical relation. And this difference in the surface tension around the droplet gives rise to a Marangoni stress boundary condition at the droplet interface. And finally, using this as a boundary condition of the Marangoni stress boundary condition, we finally model the fluid flow both inside the droplet and also outside the droplet using an unsteady Stokes equation. So we solved this entire model using a numerical scheme in COMSOL, and we can see, and now I can explain how the droplet moves due to the simulations. So what leads to the droplet motion at the very early time stages? So initially, the droplet is isotropic, and also the droplet releases chemicals isotropically from its surface. So there is no symmetry breaking. However, if there is a certain, even a finite asymmetry developed at the surface of the droplet in terms of the polymer concentration, So this symmetry is exaggerated because the diffusion coefficient of the polymer is extremely low. So as you can see, over time, the droplet develops some kind of a tail, and this actually reduces, it gives rise to this asymmetry, which is required for the droplet propulsion. So what it actually does is that it gives rise to this Marangoni flows. As you can see from the first to the third picture, that the flow moves from a region of high chemical concentration to the regions of low chemical concentration. And because of these Marangoni flow, the droplet as a unit moves forward. And these trails are exaggerated over time, and the chemical builds up over time and forming a trail because the diffusion coefficient is extremely low. And because of this, there's a continuous feedback mechanism which gives rise to the tail and sustains the tail and gives rise to Marangoni flow. And as a result, the droplet keeps on moving throughout the Petri dish. So in our simulations, we could identify the three regimes as seen in the experiment before. So initially, we also have the ballistic regime, where the droplet moves around the Petri dish. And in the intermediate time scale, we have the chiral motion. And also, eventually, we have the self-trapping of the droplet, where the motion is still chiral, but confined to a very small region. And the mean displacement of the droplets also matches with that of the experiment. Now the question is, why do the droplet go from the ballistic to the chiral regime? So for the ballistic regime, it is intuitively easier to explain why the droplet moves along the rim of the Petri dish, because initially the rate of polymer emission is extremely high and the droplet velocity is also high. And because of the circular geometry of the Petri dish, it is moving along the border and shows a circular motion along the border. But why does this, the motion suddenly goes from ballistic to chiral? So this is the question we ask. And to understand this transition, it is important to see how the chemical or the polymer concentration evolves within the Petri dish. So let's see. So initially, the droplet moves, it gives off the trail, and because the diffusion coefficient is extremely small compared to the advection, the trail is almost localized at behind the droplet. And you can see that the trail dominates, and at a certain time, there is a portion of the Petri dish along the edges where you see a dominant peak in the chemical composition. As the droplet moves through the Petri dish, you see that the trail takes the form of an annular ring kind of a structure. It cannot diffuse because the diffusion coefficient is very small. And so this trail is very long-lived. And at the later times, the droplet, when it moves throughout the Petri dish, it can interact with its own trail. So remember that the activity or the surface activity of the polymer of the droplet is exponentially decaying. So at some point of time, the amount of fresh polymers into the system decreases, and the droplet is basically governed by the old chemical, which is already in the Petri dish, that is the trail. So at some point, this trail overpowers the droplet, and as a result, the droplet slowly transitions into this kind of a chiral motion. So this is what we call the memory effect. So the interaction of the droplet with its own self-created memory, the trail, causes this kind of memory effect, which leads to its transition from the ballistic to the chiral propulsion. So what is actually happening? So this is the concentration profile of the droplet, but what is actually happening in the flow, which takes the droplet from a ballistic movement towards a chiral movement? So if you zoom in, so we look at the three stages of the droplet motion. So in the first panel, and the A and D, you see both the chemical concentration of the polymer and also the flow field around the droplet in the ballistic phase. So you see, if you see the region around the droplet in the panel D, the flow field decreases very fast as we go away from the droplet interface. But what happens after the droplet has taken many turns toward the Petri dish and is almost in the chiral region or the chiral dynamics domain? What you can see is that the flow around the droplet does not really decay so fast, and the old chemical and the fresh chemical, they kind of alter the flow around the droplet. So if you see carefully in the panels E and F, there is a vorticity developing around the droplet. And this vorticity is actually responsible for making the droplet turn and go back into this annular structure of the chemical trail whenever it tries to go out. So what happens is that the old chemical trail, because it cannot diffuse faster, it's very long-lived, it induces this memory effect. And whenever the droplet comes near this region of the trail again, the flow around the droplet is strongly modified, and this gives rise to this vortex structure around the droplet, and as a result, the droplet shows this chiral behavior. So this is also what is shown in a schematic figure over here. So initially, you have the droplet moving in the ballistic phase, shown in the green arrow, and at later times, the droplet undergoes a strong angular force or torque at the points 1 and 3, which gives rise to, which makes it turn back, and so on starts the chiral motion of the droplet. So can we try to quantify what is causing this chiral motion? Because we said that the flow around the droplet is strongly modified, giving rise to this vortical structure. So what we do is that we focus on the region around the droplet. So this is denoted by this white circle around the droplet at each time step. And we calculate the average vorticity of the fluid in this region, which is surrounding the droplet. So in the ballistic phase, the average vorticity is almost constant. It's positive, around 0.5. And this is because the droplet is slowly turning, is showing a circular motion around the petri dish. And this is a characteristic for that kind of motion. There's a characteristic value for that motion. When it goes to this chiral phase, you see that the droplet, the vorticity, the average vorticity is still positive, but it fluctuates. It shows many fluctuations. And the local maxima, as you see in this inset, they correspond to the region where the droplet turns. So what happens is that at the turning points, the vorticity is a maximum. The vorticity of fluid around the droplet is maximum. And this is what causes the droplet to turn back. So you see this kind of peak structure, this kind of fluctuations in the vorticity in the chiral regime. These fluctuations continue also in the self-trapping regime. However, the average of the vorticity is 0, which means that the droplet is basically confined to a small region, but it still shows a chiral motion because it still goes through this region of high and low vorticity, which makes it turn. We could also try to correlate the vorticity of the fluid around the droplet with the motion of the droplet itself. So in the upper panel, we plot the angular displacement of the droplet as with regards to the center of the petri dish. So in the ballistic regime in the figure B, you see that the angular displacement is linearly increasing because the droplet goes around the petri dish. In the chiral regime, it is almost showing a quasi-periodic behavior. And in the self-trapping regime, it is almost periodic and it oscillates between 2 values. So if you calculate the power spectrum of this angular displacement of the droplet and also the average vorticity, you see that in the chiral and the self-trapping regime, they correspond to the same frequency peaks, which shows that these two quantities are correlated and is a reason behind the chiral dynamics of the droplet. So in a nutshell, to summarize, so we have introduced a new type of droplet swimmer, which can show self-propulsion due to the self-freezing at the boundary. We don't require any explicit symmetry breaking for this self-propulsion to occur. And this is powered by spontaneous symmetry breaking due to the Marangoni stresses at the interface. And this droplet can exploit the memory effects due to the old polymer trails, which it itself generates to transition into chiral motion from a ballistic one. So I would like to thank all the collaborators in this project from Darmstadt and also Wuhan, and also the funding agencies for the funding. And thank you all for listening. So that's all from my side.
Okay. Thank you, Atriel. So very interesting the work. A lot of quantitative analysis on the phenomenon. So any question from our audience? So if you have a question, you can unmute yourself and ask Ariel directly.
I have a question. So very nice presentation.
Thank you.
I wonder what the effect of the container geometry is on the motions. So, you know, a circular container obviously exhibits nice symmetry, but what do you think would happen if, let's say, the container was not circular, but maybe like a square? Would you get the same type of behaviors?
Yeah, it's a good question. I think so. In the ballistic phase, the ballistic movement, it is definitely due to the circular geometry of the petri dish or the boundary, as you say. But we are not really sure if the chiral movement is due to the geometry of the droplet. But we haven't really tried with different geometries of the petri dish or the bounding region, and would be definitely interesting to try out. Yes. But we haven't seen the effect of the geometry on this type of swimmers yet.
Okay, thanks.
So any other
I have a question.
questions? Yeah, go ahead.
Then I have a question. It's a really nice talk. I have two questions. The first is, in the experiment, when the drop is freezing, does the shape change from spherical, and does that shape change have any effect? And the second question would be on the simulations. So it's more like a comment. Typically, the wake behind a cylinder is you have more vorticity when you have a cylinder compared to when you have a sphere. So maybe, do you think that is a reason why you're seeing a larger trail in your simulations behind the particle?
Okay, so to answer the first questions, thanks for the comments. So to answer the first question, the shape of the droplet doesn't really change. So as you said, with solidification, so it proceeds in a very uniform way from the edges to the interior, and the spherical shape of the droplet is still preserved. Okay. And regards to the trail, if I followed you correctly, so the trail is due to so if you see this equation from the modeling point of view, so we have this advection diffusion equation governing the polymer evolution over time. So hence, if the diffusion coefficient is extremely smaller, so this is responsible for the trail because the advection really dominates and the polymer really can't diffuse extremely fast. So I think you asked about why the trail forms. So I would say the trail is forming because of a very small value of the diffusion coefficient in these polymers.
So yeah, thanks for clarification. So on the second point, what I was trying to say was maybe you're seeing a larger trail since the simulation is 2D. Is that possible? Because you have generally when you look at flow behind a sphere or a cylinder, you have more vorticity behind the cylinder at a given Reynolds number. So I was thinking maybe you have a geometric effect in the simulation. Okay,
that's a good point. Yeah, we haven't really performed the simulation in 3D. But yeah, I mean, this is a good point I'll keep in mind, so we can check this if this is a reason for an enhanced trail. Yeah, due to the symmetry of the droplet, maybe. Thanks for the comment.
Okay. Yeah. Thanks.
Oh, hi, Arija. Very nice talk. So I just have a question about your experiment. So I'm just wondering, how do you measure the chemical concentration in your experiment? So you show the concentration change during the dynamics. So just out of my curiosity, how you can measure the concentration?
So you mean the polymer emission? So this curve here, how it
you have a video shows the chemical concentration change if you move forward.
I mean in our simulation, I think. Okay.
No
Assuming this.
simulation.
So this one?
Oh, yes. Oh, this is the
Okay.
simulation, right?
This is a simulation. Sorry, it
Oh,
was not clear.
oh,
Yeah.
sorry.
So this is not
So
from
I
the
thought
experiment. Yeah, this is from the simulation. Yes.
Okay, I see. Yeah, thank you.
Yeah, but from the experiment, we can measure the flow field. Yeah, but we didn't really measure how the polymer evolves. Yeah, so this is from the simulation point of view. Yes.
Okay. Yeah, thank you for the clarification.
No problem. Thanks.
Any other questions?
I have one last question, Zen.
Sure.
So in your computational model, the empirical model you use for the surface tension,
Mm-hmm.
yeah, down here on the lower right.
Yeah. Sorry.
Is that a classic way to define the surface tension, or are there other models to do this? I mean, obviously it's a decreasing function with increased concentration.
Yeah. So
I'm just wondering if there's any other models that are, I'm sure they'll give the same qualitative behavior, but maybe
so we
quantitative.
actually tried also a linear model. So I think the most common model is to take a linear function, which if we increase the concentration, the separation decreases. And but we took a more general approach by using this formula. So in a very least order, you can see that it gives back the linear approach, the linear model. But we took the more empirical formula for our observations. Yes, to account also for the nonlinear terms.
Okay. Thanks.
So in your model, so the droplet, when the droplet is moving, do you consider the deformation because it's a droplet on the surface, right? Do you consider the geometry change?
No, actually. So the geometry is assumed to be spherical. It's keeping the shape. And also in the experiment videos, we saw that the droplet almost preserves the shape. And there's a reason we, in our simulation, we simplify saying that the droplet doesn't really change the shape and there's no deformation.
Okay, so do you think that geometry will play a role in the motion, or
sorry?
maybe no? Do you think if you consider currently you consider a spherical droplet, right? So for the active motion, do you think that the geometry also play an important role? For example, if later I change another kind of shape, maybe I use other driving mechanism. It's not by the surface reaction. I use some solid geometry, but use different shape. Do you think this will change the motion?
Sure. I think it will. I mean, if there is a solid or any modify, if you modify the shape of the droplet, and it probably would give rise to chiral motion in itself because the geometry also has an effect. So the idea here is we start with a really isotropic spherical shape, and we can still generate this kind of a behavior of chirality. But it is really possible that we change the shape of the droplet, or not the droplet, but of a solid, let's say. And it is possible to generate different kinds of motion, due to it. So
yeah, usually if you use solid kind of objects, this will usually generate a 3D motion, right? So I'm not sure because in your experiment or simulation, you kind of fix the droplet the motion on the 2D plane. I'm not sure in the real case if you have some active particle in the bulk in the solution environment, if this kind of pattern still reserved, or it will generate a significantly different pattern.
You're right. I mean, even in this experiment which we have, it's actually in a 3D experiment. Also, it's a 3D scenario, right? And but we only consider a 2D surface, only the disk surface, as you say. But we could capture all the necessary ingredients with this 2D model. But I mean, if we increase the complexity of the model to do very realistic 3D simulations, I think we will still get this the similar behavior. of the droplet, but with different effective coefficients like the surface tension constant and other things which were used to fit the data.
Okay, thank you. So any other question from our audience? So if there's no more question, then we will end here today. So if you have additional question, you can contact Dr. Mukhopadhyay.
Mukhopadhyay. No, you did it perfectly.
Directly. Okay, so then we'll end here today. Thanks everyone for your attending. Bye bye.
Thank you. Bye.